Yeah.
So today we're going to do lecture 4, and what our goal is to look at the control of the digestive system kind of from a homeostatic standpoint, and then to talk about the enzymes and their activity within the digestive system for digestion of macromolecules. So I wanted to kind of start with a review of the parts of the digestive system and kind of the idea of homeostasis. So recall homeostasis is a dynamic state that's brought on by the nervous system and the endocrine system. And so when we're thinking about the nervous system component, then remember that as we're looking at the wall of the digestive system, we actually have layers. And so the internal layers of the digestive system are the mucosa, and the epithelium is the innermost layer. And so it's within the epithelium that we're going to have specialized cells that we call enteroendocrine cells that are going to be the hormone component to homeostasis. As we go outward from the mucosa to the submucosa and to the muscularis, then remember that when we looked at it in lab, that within the submucosa we have this submucosal plexus or the plexus of Meissner's, and then in the muscularis layer we have this myelenteric plexus and the plexus of Auerbach. And so these two plexuses are the autonomic nervous system's control of Okay, so what happens is the submucosal plexus, or the plexus of Meissner's, is a plexus that's going to control the processes of the secretion of glands like submucosal glands and stuff that are part of the digestive system. The myelenteric plexus, or the plexus of Auerbach, is actually going to control peristalsis and mass peristalsis within the digestive system as well. And so what we're going to focus on more is the role of the enteroendocrine cells that are part of the epithelium and the role of those hormones at trying to maintain homeostasis within the digestive system as well. So what I've done is I've created charts for you that summarizes all of the important components of the hormonal control. And so I've tried to set them up in a pattern where they're set up similarly so you can think about it in regards to a process. So at the top there'll be the name of the hormone, for example on this one, gastrin. And then underneath it are 3 subheaders. So where the hormone is secretion, it is the secretion component. What causes the hormone to be secreted is the stimulus component. And then the action of the hormone in regards to homeostasis within the digestive system is under the action. Okay. So gastrin is secreted by enteroendocrine cells in the bottom of the gastric pits that we call G cells. What initiates the release of gastrin is boluses of food entering the stomach and causing the stomach to be stretched. And also as food enters the stomach, the gastric pH is going to elevate, and so that's going to cause the release of gastrin. So one of the primary roles of gastrin then is to target the parietal cells that produce hydrochloric acid and increase their production of hydrochloric acid. And then the role of the stomach is to mix the hydrochloric acid and the food material to eventually liquefy it. So it's also going to increase gastric motility or peristaltic contractions within the muscularis of the stomach. It also stimulates the growth of the gastric mucosa. So remember, the epithelial component of the mucosa are simple columnar cells, and we constantly have to replace those simple columnar cells to make sure that our gastric lining is in good shape. And hydrochloric acid tends to damage the gastric mucosa. So what we want to do is stimulate the continued replacement of cells to maintain a healthy environment in our stomach. Also, because as we increase gastric motility, and the food becomes more liquefied by the hydrochloric acid, it would tend to be pushed up into this esophagus more. That gastrin also causes a contraction of the lower esophageal sphincter to try to prevent gastric reflux from occurring. As the food is being liquefied, the liquefied components are moving into the antrum of the pylorus. So we're also going to initiate the process of emptying the stomach. So we're going to relax the pyloric sphincter so that as the liquefied food enters the pyloric antrum, it can be moved into the small intestine. And then the other day we were talking about a gastrocolic reflex, and a gastroileal reflex, where as we put food in our stomach, we want to begin to make sure that the lower part of the small intestine has emptied into the large intestine, and then eventually the material in the transverse colon is moved quickly into the rectum, so that we have room for the material that needs to be processed that's going to be vacating the stomach. So it also relaxes the ileocecal sphincter so that the material in the ileum can move into the cecum and the S in the colon. So what's really going on with this homeostasis is we're creating a hormone that not only is active in this case in the stomach and controlling the activity of the stomach, but it also controls the activity of, for example, the small intestine, so that the two organs are working together in regards to doing the digestive process. So the next hormone we're going to look at is a hormone called somatostatin. And then it's secreted by endocrine cells called D cells in the gastric pits of the mucosa of the pylorus of the stomach, so the tapering part of the stomach itself. When we did the stomach initially, we didn't talk much about somatostatin. And so what we're going to see is that it's a hormone that interacts with the last hormone that we just talked about, which is gastrin. So the stimulus for somatostatin is that it's actually being continuously released. So it's a hormone that's being released on a continual basis. And what the hormones are going to be done, it's an antagonistic hormone to gastrin. So when gastrin is present, it overrides the hormone. Plus we have the submucosal plexus involved in the situation. So when we think about it, the hydrochloric acid in our stomach is critical to the health of the stomach. So if we have too much hydrochloric acid all the time, then it promotes the erosion of the mucosa of the stomach. So when food isn't in the stomach, then we don't want hydrochloric acid being produced. So somatostatin is being produced all the time, and then that somatostatin is limiting the production of hydrochloric acid. So as the stomach becomes distended, then we're going to get the release of gastrin, and then gastrin is actually going to override somatostatin, and then we're going to get an increased production of hydrochloric acid. As the stomach empties and the level of gastrin drops, then the action of somatostatin is to prevent gastrin from being produced when the stomach is empty. So the two hormones kind of work together to maintain the appropriate environment within the stomach for the digestive process. So the next hormones we're going to talk about are actually produced by the small intestine. And what we're going to see is that their stimulus is the movement of material chyme from the stomach into the small intestine. So secretin is a hormone that's secreted by endocrine cells in the crypts of Lipids in the small intestine. So remember, those are the downward folds that we see characteristic within the small intestine itself. Okay? And then the stimulus is going to be the movement of chyme into the small intestine, and in particular the movement of acidic chyme into the small intestine. So the homeostatic point for the small intestine is to have a pH that's slightly above 7, and so as this acidic chyme enters the small intestine, the pH is lowered, and that triggers the release of secretin. So what secretin is going to do is have an impact on accessory organs and parts of the digestive system to try to bring the pH back up to that homeostatic point that's a slightly basic pH. So one of the things that secretin is going to do is stimulate the secretion of pancreatic juice and bile juice. So remember, pancreatic juice is a product of the eosinophil cells in the pancreas, and bile is a product of hepatocytes in the liver. And what they do is they produce bicarbonate ions, and then bicarbonate is a very important buffer. So as this acidic chyme enters the small intestine, then we're going to get the increased secretion of pancreatic juice and bile and the movement of bicarbonate into the lumen of the small intestine to act as a buffer. Also, because as chyme is exiting the stomach, it's an indication that the digestive processes in the stomach are going to start coming to a conclusion and the stomach is empty. Then secretin is also going to inhibit the parietal cells in the gastric pits and decrease the production of hydrochloric acid to help maintain a healthy stomach mucosa. It also promotes the growth and maintenance of the pancreas. So since the pancreas's role is very important in the production of these bicarbonates, then secretin also helps maintain the center cell populations within the pancreas. And it acts interactively with the next hormone we're going to talk about, which is cholecystokinin. And what we know about hormones is that there's a permissive effect within hormones, and then there's a synergistic effect within hormones. And the two are where one hormone precedes another, increases its activity. That would be permissive. And then if the two hormones are together and that increases its activity, then that's synergistic. And so secretin is synergistic with cholecystokinin, enhancing its effects as we go through those. And then it also increases secretin increases the rate of bile secretion by hepatocytes, which links back to number one, which is that need to increase bicarbonate presence in the small intestine, the duodenum in particular, for buffering the acidic chyme that's coming down. Okay. So now we're going to talk about CCK, cholecystokinin, which is created again by cells in the crypts of Leverkuhn within the small intestine. And so they're enteroendocrine cells again. So the pattern that we use is we take the subunits of the hormones, so C for chole, C for cysto, K for kinin, and then we create an acronym for the hormone, so CCK. And then, so these are CCK cells. And then, again, it's going to be the movement of chyme out of the stomach into the small intestine that's going to facilitate the release of CCK. But instead of being the acidic properties of chyme that it was in secretin, it's going to be chyme-rich in the initial breakdown products of proteins, amino acids, the presence of fat in the form of triglycerides, and the presence of some breakdown products from fat digestion, which would be fatty acids that are in the small intestine. And so as this chyme, rich in these nutrients, is moving out of the small intestine, then when CCK is released, its name is in reference to its activity on the gallbladder. So what was learned very early on is that it caused gallbladder contraction. So hence its name, cholecystokinin, is a reference to acting with the gallbladder. And so that's one of its actions. It also, because digestive enzymes are secreted by the pancreas and dumped into the duodenum, then it's also going to stimulate pancreatic juice that's rich in digestive enzymes. So a combination of secretin and CCKs simultaneously results in pancreatic juice that's both high in bicarbonate and high in digestive enzymes. And then recall when we talked about the ductal wire sum and the common bile duct, that they come together to form a hepatopancreatic ampulla. And then this ampulla extends a little bit into the wall of the small intestine, which we call the duodenal papillae. And where the papillae is, we have a sphincter that controls the release of both bile and pancreatic juice. So CCK also opens the sphincter of Oddi so that this fluid rich in digestive enzymes exits the duct and goes into the small intestine where it's going to be active. And it's not just retained in the duct where it would begin to digest the wall of the duct. And then a contraction of the gallbladder to move bile, which it will enhance our ability to digest fats. And then it also inhibits gastric secretion and motility. So as the stomach's emptying again, CCK with secretin is going to begin to decrease the activity of the stomach and move the activity into the small intestine. Then there's some research that suggests that CCK also targets the hypothalamus in the brain. And that may be the connection that our brain uses to know that we have this digestive material moving into our small intestine. And so it reduces our hunger feeling and begins to stop us from continuing to ingest food. So that's kind of a nice connection to that process. And probably why there's a suggestion that the slower you eat, the less likely you are to overeat in terms of the amount of calories that you take in and maintaining the overall homeostasis of body weight. All right, so another hormone that's produced within the crypts of lubricant in the small intestine is gastric inhibitory peptide. So following the same pattern again, first letter of each component, so GIP. And then it's named beautifully because it tells you what it's going to do. It's going to be inhibitory to the stomach. And so again, it's going to be the movement of chyme out of our stomach into the small intestine. And you're going to see it kind of overlaps with CCK to some extent, in that it's going to be chyme rich in fats that we just talked about, and fatty acids that was similar to CCK, but then also carbohydrates this time. So the presence of glucose entering the small intestine as well. So with those two hormones, we secrete a hormone that's going to affect the activity for the overall digestion of those macromolecules. And glucose is really the key to this first action. So if we have glucose present in chyme, then we're going to begin to absorb it through the ileum. And then the sugar is going to end up in our blood. Well, to effectively carry sugar in our blood and to effectively have cells recognize the sugar and take it up as a resource that can be used to make ATP, we have to have insulin. And then insulin is secreted by beta cells in the islets of Langerhans and the pancreas, which we're going to talk about in a little more detail in a minute. So the presence of sugar is key to stimulating the release of insulin by the beta cells, so that now that we have sugar in our small intestine, we can manage its transport and uptake by cells in our blood. It also inhibits gastric secretion and motility again. So going back to the idea that the stomach is emptying so we can decrease its activity. It stimulates lipogenesis by adipose tissue. So we have a bunch of words that we use when we're thinking about biochemical processes. So anytime the word ends in genesis, it means we're going to take small molecules and make large molecules from them. So the first part of the word is used as a reference to the molecule that's being produced. So lipogenesis is the production of fats and by adipose tissue itself. So the fact that we have triglycerides and fatty acids that are going to be absorbed and digested in the small intestine means that we're going to have this new input of fat into our blood. And as that's carried to adipose cells, and it stimulates the lipogenesis to form and store fat molecules. Also, if we overeat and we have abundant glucose, where we have all the glucose needs that we need in terms of the formation of ATP, and we've increased our glycogen stores within skeletal muscle and liver, then we can also use glucose to make, through lipogenesis, to make fats. And so it promotes that retention of these macromolecules for future energy needs. And then it stimulates glucose use by skeletal muscle. In other words, we're going to take the additional glucose that we aren't using for ATP and convert it to glycogen within skeletal muscle to store it, as well as facilitate the use of ATP within the muscle material as well. All right, so we have GIP, now we have VIP. And again, its name is going to help us understand what it does. So vasoactive intestinal peptide is another hormone produced by the crypts of Lieberkühn within the duodenum and the small intestine. So again, vasoactive V, intestinal I, peptide P. So peptide tells us this is a protein molecule, and it's released when chyme enters the small intestine. So it's not really the fact that we have different properties of the chyme, it's the distension that's created by movement of chyme into the small intestine. And then what VIP does, obviously, is it causes vasodilation. So it helps regulate blood flow to critical areas. So if we increase our blood flow to our liver and our pancreas, and actually increase blood flow within the wall of the small intestine itself, then we can stimulate the production of bicarbonate at a higher rate to act as a buffer system. It also is going to inhibit gastric secretion by causing vasoconstriction of blood supply to the mucosa of the stomach and decrease resources available to G cells. And then it dilates intestinal capillaries. And then that's a real critical role that we're going to end this lecture with. And that's the idea that as this product, chyme, is moving through our small intestine, we're going to go through the process of digesting macromolecules into the building blocks that we need for our body to make new resources. And what we're going to do is we're going to move them through the intestinal mucosa into blood vessels in the underlying lamina propria, which is the areolar connective tissue, as you'll recall, that occurs underneath the mucosa. So we want to, if we're not involved in absorption, then we don't need these capillary beds to have as much blood flow. If we're involved in the process of absorption, then we want to increase blood flow to these capillary beds. And then we're actually going to kind of wait until the third unit, which is where we do blood movement in the body. And we'll talk about how we alter blood flow through capillary beds at that point in terms of managing the capillary load of blood. And so we'll come back and revisit the mechanism in the third unit. All right. So what I'd like to end with this part of the lecture on hormones is moving away from the digestive system proper, the stomach and the small intestine, and move to one of the very important accessory organs for the digestive system, which is the pancreas. And so we've already talked about the fact that the pancreas and the acini of the pancreas produce bicarbonate and digestive enzymes. And so what we want to do is look at islands of cells within the pancreas that we call islets of Langerhans, and they're endocrine cells. So it's the endocrine component of the pancreas. So when we look at islets of Langerhans, we can identify 4 cell populations: alpha cells, beta cells, delta cells, and F cells. And each of these cells is going to be responsible for producing a different hormone. So if we look at the first 2, alpha and beta cells, then they are going to be involved in helping control blood sugar levels. So alpha and beta cells are cells that we're going to use to control blood sugar. So what happens as the digestive process occurs is that we break down complex carbohydrates like starch and glycogen and disaccharides like sucrose and maltose and fructose. And then what we're going to do is we're going to absorb the simple sugars, which is glucose. So via the absorptive process, we're going to get that elevation in blood sugar. And as we've already talked about, we need to have insulin that aligns with the sugar so that cells can take it up. So the key to beta cells is that they produce insulin, and insulin has to be bound to sugar for our cells to pick up the sugar and to use the sugar for metabolic processes. So insulin is involved in decreases of blood sugar. So the way to think about it is, as we're absorbing the sugar through our gut lining, our blood levels of sugar are going to elevate, and we're going to enter a state of hyperglycemia. And then it's insulin that brings us back out of that state of hyperglycemia by lowering our blood sugar back to normal levels. Okay? So if we haven't eaten for a long period of time and our cells are active metabolically, they're going to use up sugar. So as we use up sugar within cells, then sugar from the blood is going to continue to be absorbed by the cells, and our blood sugar levels are going to drop. So we would enter a state of hypoglycemia in periods of time where we're using up sugar but we aren't absorbing sugar. And so as blood sugar levels drop, because their levels are critical to the metabolic processes, particularly of our brain and of myocardium, then we're also we'll have alpha cells secrete glucagon. And then glucagon is going to cause the liver and skeletal muscle to release stores of sugar from glycogen and return the sugar back to our blood. So basically the general trend for glucagon is to increase blood sugar level. So the two hormones together help us maintain sugar balance in our blood and maintain homeostasis. So just remember that insulin is a hormone released as the body enters the state of hyperglycemia, and glucagon is a hormone that's released as the body enters states of hypoglycemia. So F cells and delta cells are going to produce hormones that are involved in different processes. So delta cells secrete somatostatin, and somatostatin is a hormone that targets both alpha and beta cells, and so it decreases the production of glucagon and insulin, and so it actually prolongs the rate of nutrient absorption and decreases depletion of nutrients in our body. And then what happens is somatostatin is overridden when insulin and glucagon levels elevate because of a drop in blood sugar. So these three kind of interact with each other. And then F cells secrete pancreatic polypeptide. And what it does is it inhibits the secretion of digestive enzymes and inhibits the contraction of the gallbladder. So this would be the hormone that would be critical between periods when your digestive system's not active because you haven't ingested food. So as you ingest food and the food moves into the small intestine, then remember secretin and CCK were involved in controlling the activities of the digestive enzymes of the pancreas and of the activity of the gallbladder. So again, if you have nothing in your small intestine that you need to digest, then you don't want digestive enzymes being secreted, and you don't want bile moving into the small intestine to emulsify fats. So pancreatic polypeptide would inhibit that activity. As chyme from the stomach moves into the small intestine, then we get the production of CCK, and then that would override this hormone and begin the digestive process. So you can see how these hormones play out with each other to try to maintain homeostasis within the digestive system, promote an increase in activity during that period of time when we're doing digestion, and then promote a decrease in activity during periods of time where we're not actively involved in the digestive process. So now we're going to move our attention to the digesting process itself. And that is looking at a set of protein molecules that we call enzymes that are involved in the process of digestion. So within carbohydrate metabolism, we have this wonderful kind of relationship between the substrate and the enzyme and the nomenclature or the naming system that we use for the sugar and the enzyme. So if we look at the proper name for sugars, lactose, the sugar in milk, sucrose, the sugar that we use, which is table sugar, maltose, and then amylose, which is a technical name for starch. And notice that all of these sugars end in OSE. So a naming pattern for carbohydrates has been to name the sugars ending in OSE. And then by convention, we name the enzyme that interacts with the sugar with the ending ASE. And then it makes it easy to connect these enzymes to the enzyme itself and then the product that it's going to digest. So we have lactase, the enzyme, will digest lactose, the sugar. So carbohydrates are really nice. So what I've done is I've created a bunch of tables to help you kind of focus on this process and also guide your focus in the process in the way in which I expect you to know the material. So we want to know the name of the enzyme. We want to know the source of the enzyme, where it's actually being produced. And then we want to know what the substrate of the enzyme is, what the enzyme is going to digest. And then we want to know the end products of that chemical process of digestion. So as you're reading this chart, it's a linear relationship. So we have the enzyme lactase produced by brush border cells, which are the microvillated cells at the top of villi. And then those brush border cells that produce the lactase help us digest lactose, the sugar we get from milk. And the end product would be glucose and galactose that then we would absorb through the gut lining and move into our blood to begin the digestive processes. So as we're looking at these enzymes, then what we have are very large macrobiological molecules called starch, which is the way we get complex carbohydrates from plant material. We could also add glycogen, which is the way we get complex carbohydrates from animal material. And so amylase is produced in salivary glands, begins the process in our stomach, and then it's reintroduced in the small intestine, in the duodenum, via the pancreas to help us digest starches. So what happens is that starches are long chains of glucose molecules, and the enzyme amylase, if given enough time to digest, is going to digest it into disaccharides called maltose. So we're not going to get complete digestion with amylase. If the transient time in our digestive system is fairly quick, then we're also going to get fragments of starch that we call dextrins. So coming out of the stomach, we're going to have maltose and dextrins as a mix, as well as starch that's not been digested. So in our small intestines, we're going to continue the use of amylase from the pancreas, which is eventually going to be able to break starch down into maltose if given enough time. To facilitate that process, we have a brush border enzyme called dextrinase whose substrate is dextrins, and that's going to help us break the fragments of starch down into glucose. And then we have maltase, which is a brush border enzyme that's going to help us digest maltose, a disaccharide, and break it down into glucose. So just join us. If you want to study, just go ahead. We're just doing a lecture. So at the end, we end up with glucose molecules that we're going to absorb through the gut lining. And then our other sugar that we come in contact with through food material is sucrose, which is the sugar you buy in bags in the grocery store. And so we have to have an enzyme that digests that disaccharide, sucrase. It's again a brush border enzyme, meaning that it's only active in the small intestines. So by the time the sucrose gets to the duodenum, then that's where we're going to begin to break it down. And then we break it down into two simple sugars, glucose and fructose, that we can absorb. And so the common pattern across here is the glucose, which is our body's number one choice for creating ATP via cellular respiration. Then we have a couple of other sugars, fructose and galactose, that we have to modify a little bit before we can actually generate ATP out of those. And we'll talk a little bit of that in lecture 5 when we talk about metabolic processes. So fats typically are ingested in the form of triglycerides, although we do get some monoglycerides and diglycerides. So remember that a triglyceride is a complex molecule made from a 3-carbon molecule that we call glycerol. And then attached to the 3-carbon molecule, we have these long carbon-carbon molecules that are these long chains of carbon and hydrogen. So we call those fatty acids. So if our fat has 3 fatty acids on it, we say it's a triglyceride. If our fat only has 2 fatty acids on it, then we call it a diglyceride. And if we only have 1 fatty acid on our glycerol molecule, then we call it a monoglyceride. So what we're going to do is we're going to take these more complex molecules and ultimately be able to digest those into this molecule, which would be a monoglyceride. And then we would have 2 free fatty acids that have been the product of the digestion. So notice that in our product column right now, we have monoglycerides plus fatty acids. So just remember that for each triglyceride we started with, we would end up with 1 monoglyceride, 2 fatty acids. So we would end up with 2 of these for every monoglyceride that we started with. So triglycerides belong to the macromolecule group that we use called lipids. And so the enzyme itself is getting into an ASE, telling you it's an enzyme, but light is going to be the first part of it. So lipase is the enzyme itself. And we actually initiate this process in our mouth with lingual lipase. We continue in our stomach with gastric lipase, and then we complete the process in the duodenum because the pancreas produces pancreatic lipase. And then it's designed to digest triglycerides into fats. And gastric lipase is probably more active in infants that are on a liquid diet of milk because it specializes in the digestion of particular fats common in milk or butter fats. And so the end product then is monoglyceride fatty acids that we're going to aggregate, and then we're going to talk about this a little bit, and then absorb into our blood so that we can use these fats for producing new membranes and for doing other things within our body. So the macromolecule that's the hardest to digest are proteins. And so we have the widest array of enzymes involved in protein digestion. And we've probably understood protein digestion for a much longer period of time. So we have an old naming system and a new naming system intertwined within these digestive enzymes that digest protein. So again, following it is we're going to have the enzyme, we're going to have where it's produced in the body, and then they're going to break down proteins. And then if we can completely break down a protein, we're going to end up with an amino acid. Fragments of proteins are called peptides. And so we're going to end up with a pattern of fragments of proteins plus free individual amino acids. Now, proteins are problematic because protein enzymes are problematic because proteins are a structural component to tissues and cells. And so we want to be very careful about where protein enzymes are active. So for the proteins that are enzymes that are produced in the stomach and in the pancreas, we're going to create them in an inactive form, and then we're going to activate them. And that's to assure that they're active in the lumen of the digestive system. So to convey that information, in the older naming system, the inactive form of the enzyme starts with OGEN. The active form of the enzyme, then we just drop the word OG, the ending off of. And then in parentheses is the active agent that activates the enzyme. So we start with the stomach, and the chief cells in the stomach, the chief cells technically produce pepsinogen. As the pepsinogen passes out of the gastric pits up into the lumen of the stomach, it's mixed with hydrochloric acid, and the hydrochloric acid changes the shape of the molecule, which activates it into the active form of pepsin. And pepsin only cleaves the bonds between certain amino acids. And so since it only cleaves the bonds between certain amino acids, the end product may be some free amino acids and fragments of proteins that we call pepsins. Peptides, okay. All right, so that's the hormone, the digestive enzyme active in the mouth. And we start no protein digestion in the mouth. So now as the chyme vacates the stomach and enters the duodenum, then we're going to have a whole series of digestive enzymes that are active in the small intestine of the duodenum. So one of the important ones is the enzyme trypsin. It's actually produced by center cells in the pancreas. And then what travels down the ductal wire sum and it ends up in the hepatopancreatic ampulla and being dumped into the small intestine is an inactive enzyme called trypsinogen. We're going to have an enzyme that's actually a brush border enzyme produced by the cells that line the duodenum, which is enterokinase. And then enterokinase is going to activate trypsin. And then trypsin will digest proteins at unique bonds between certain amino acids and give us more free amino acids and fragments of proteins called peptides. And what's kind of cool is that the active enzyme trypsin is going to be the activators for some other enzymes. So that it'll actually help us activate enzymes. So another enzyme that we produce in our small intestine is called chymotrypsinogen. And that's what's produced by the center cells of the pancreas. Again, travels down the ductal wire sum through the hepatopancreatic ampulla to get into the lumen of the duodenum. Once it's in the lumen of the duodenum, then trypsin activates it to chymotrypsin. Chymotrypsin, again, is going to digest proteins, but between certain amino acids. So it, again, is going to fragment proteins into peptides and may provide us with some free amino acids. So all the hormones that we see on this first part of this protein digestion chart are going to fragment proteins and maybe give us some free amino acids. So as we talk about the additional digestive enzymes for proteins, what we're going to see is we need a wide array of digestive enzymes to end up with all amino acids. And that's why protein digestion requires such a large array of enzymes. If you took a biochemistry class, you could actually look up on charts to see which amino acids each individual enzyme listed on this list is going to work between, which would explain the type of fragments that we would actually create. So one of the proteins that is common in connective tissue and areolar connective tissue in particular is elastin. And so we actually have an enzyme that helps us break down the protein from connective tissue. So it's an enzyme that's been discovered more recently, so therefore its nomenclature follows the more recent pattern of naming enzymes. So the enzyme ends in ASE. Well, if the enzyme ends in ASE, it becomes problematic using the old nomenclature system of ending the inactive enzyme in OGN. And so what we do is we add a pro in front of the inactive form. So what's actually produced by the pancreas and the inner cells in the pancreas is proelastase, and then trypsin is going to convert it to the active form of the enzyme in the lumen of the duodenum, elastase. Elastase is going to work on proteins, but more specifically a specific protein called elastin. And then elastin is going to be fragmented again into fragments called peptides, and then amino acids. And then some people use poly in front of peptides to show that they're fairly large molecules themselves. So one of the kind of cool enzymes that we have, which is one of the first enzymes that we're going to talk about that actually has the capacity to completely digest a protein, is the enzyme carboxypeptidase. So it's produced by the pancreas again in an inactive form, pro-carboxypeptidase. And then trypsin converts it to carboxypeptidase that actually then begins to break down the protein. But it breaks down a protein in a very particular pattern. And so to understand that, what we have to do is we have to look at the chemical pattern of a amino acid, the structure of an amino acid, and then recognize how amino acids are put together to form a protein. So the basic chemistry of an amino acid is a building block of carbon, nitrogen. And then if it's a free amino acid, we would have two hydrogens attached to the nitrogen. And that is an amine group. And hence, that's where the name amino comes from in an amino acid. The first carbon over here is a carboxylic acid group. And so it is our acid group. And it tends to want to denote a hydrogen ion here. And so that's where the name amino acid comes from, is the fact that we have this compound that's got an amine group on one end, carboxylic acid group on the other end. And then what makes amino acids different is a functional group that we can refer to as a R group. And that could be as simple as a hydrogen ion to as complex as a cyclic carbon. And in biochemistry, you could be expected to know all those different amino acids. And so what happens, and if you kind of recall a pattern that exists from discussions of DNA, is that DNA is a language of three-letter words called codons. And then a codon codes for a particular amino acid. So it's the information in our DNA that allows us to make enzymes, which are proteins, by putting together a particular sequence of amino acids. So what would happen in our ribosome is that we would have two amino acids that would get combined. And the way we combine it is we essentially remove a water molecule. So what we end up doing by combining the two amino acids is removing a water molecule and creating a bond between the nitrogen and carbon. So we end up with a sequential pattern within a protein of carbon-carbon nitrogen, carbon-carbon nitrogen, continuing for the entire length of the protein molecule itself. So then what the enzymes that we've been talking about have to do is add the water molecule back to split these amino acids back up. Well, the enzyme we're talking about, which is this carboxypeptidase, is going to recognize this end of a protein molecule. And so it'll recognize this carboxylic acid. And then it's going to begin to break the protein molecule down from one end, continuing through the next end. And if given enough time, then carboxypeptidase would result in the end product of all three amino acids. So this is actually the first enzyme we've talked about where we don't get fragments of proteins based upon the way in which the enzyme breaks down these peptide bonds. And given enough time, we're going to completely digest a protein. So the next one, enterokinase, is actually a brush border enzyme. So it's produced by the microvilli cells. So it's actually produced to the lumen of the duodenum. So we aren't transporting it down a duct, so we don't have to worry about where it's active. So it's an enzyme that does not come in a non-active form. So it's an active form when it's produced. And then remember, enterokinase's primary role in the whole process was to activate trypsin. So that trypsin can activate the sequence of other enzymes that are required. So the one take-home message is that all of the proteolytic enzymes that are produced where they have to travel through the gastric pit, as in pepsin, and or move down the duct of Weyerson to get into the lumen, are all going to be produced in an inactive form and then activated. But the the enzymes that are actually produced by brush border cells are secreted directly to the lumen, and they're not going to have an inactive form. So we just have a couple of more protein enzymes to actually look at. So aminopeptidase is an enzyme like carboxypeptidase that can completely digest a protein. And it gets its name because it recognizes our amine group here. So it starts at this end of a molecule and digests the molecule in the opposite direction. So again, if given enough time, because it's recognizing the terminal amino acid with an amine group, then we're going to end up with simple amino acids as our end product. And then we end up with a lot of dipeptides, which is what I drew up here, which are two amino acids held together. So we actually end up with an enzyme that can digest dipeptides, which is dipeptase. And it is, again, a brush border enzyme, so it's not going to have a precursor form, and it's going to end up with the production of individual amino acids. So carbohydrate digestion had a number of enzymes involved. Lipids, primarily one enzyme, lipase. And then when we get to proteins, we have the largest group of enzymes to help us facilitate the digestion of proteins. And then proteins are just so fundamentally important to our body because they're a structural component of the cells. They moderate biochemical reactions by producing enzymes, and so that kind of makes sense. So our last group of macromolecules that we ingest are nucleic acids, which remember are DNA and RNA. And so DNA and RNA encodes information that we pass from cell to cell to cell in formation of new cells, and then from individual to individual in the reproductive process. So because the DNA sequences are unique, then we don't want to incorporate DNA from other material like animals, plants. So what we want to do is we want to break down the DNA into its building block. So basically, the building block for DNA, as you'll recall from general biology, is a nucleotide. And then we would share common nucleotides between RNA and DNA. So both DNA and RNA would contain A, adenine, both contain G-guanine, and then they would both contain C, which is cytosine. So we would get those common from both. So the difference between DNA and RNA in terms of nucleotides is that within RNA we would have uracil, and then within DNA we would have thymine. Those are the differences in the two. So what we end up doing is taking in RNA, and we take in DNA from the cellular material that we ingest. And DNA and RNA actually have to be processed by acid before we can actually digest them. So we have no digestion occurring in the mouth and the stomach, and we have to use hydrochloric acid to help this process to end up at a point where we can begin to digest DNA and RNA. So the enzymes involved, RNase and DNase here, are both the center cell products, so they're produced by the pancreas. And so DNA digestion only occurs in the duodenum and the small intestine by these enzymes. And what we're going to end up with then is this nucleotide, which remember is the sugar, ribose or deoxyribose, with a phosphate attached to it, and then this nitrogen base, adenine, guanine, cytosine, thymine, or uracil. And then we also have a brush border enzyme, which would be produced by the cells lining the small intestine again, that breaks down nucleotides. So it's called the nucleosidase, and it separates the phosphate from the sugar pentose, which is deoxyribose or oxyribose, and then the base, nitrogen base, A, C, T, or G. Then that allows us to absorb all these end products and manufacture our own DNA out of it without incorporating genetic material from other organisms, which is critically important. You can give that some thought. So I want to finish the lecture moving into the ileum and thinking about the end products that we just created with all these enzymes and how we move those through the mucosa of the small intestine and into the blood. Right? So if you think about the two processes by which we move material across the membrane, the free process is diffusion. But the problem with diffusion is it always requires what?
Water.
A concentration gradient, going from an area of high concentration to an area of low concentration. So in the digestive system, as we would try to absorb by diffusion, the more we absorb, the concentration gradients become closer and closer together, and the rate of diffusion would slow down. So it would be very problematic to be able to absorb all of the end products of digestion by using diffusion. Plus we have the chemistry involved in the bilipid layer of the membrane and the size and shape and charge on a molecule on whether it can pass through membrane very readily. So if we want to untie ourselves from diffusion gradients and we want to facilitate the movement through a membrane where it might be problematic otherwise, then we use active transport. And active transport requires us to use energy. So when we talked about active transport in 241, we talked about kind of three different mechanisms. A uniporter, where a molecule is actively transported by itself, and then coupled transport. And we had molecules being moved in opposite directions, and antiporter, and molecules being moved in same directions, a symporter. So what we're going to see is that we're going to depend upon active transport to actually move molecules. And that unties us from a concentration gradient. It resolves the chemistry involved between the plasma membrane and the size, shape, and charge on molecules. And so to facilitate the absorptive process is why the small intestine is the only organ with villi, which are these finger-like extensions that extend into the lumen. And our absorptive cells at the top of the villi are going to be microvillated, which increases the absorptive surface area of those cells. And then what we're going to do is we're going to move the material that's passing through these microvillated cells into capillary beds that extend up into the villi. So these villi capillary beds. And so not only do we increase the mucosa for the absorptive process by increasing the number of these microvillated cells with villi, but we also increase the total capillary investment in the wall of the ileum by having these capillary beds extending up into the villi itself. So what we have over here then are cells, and then this jagged pattern is to represent the villi that we're seeing up there. And so what we're going to do is look at moving a molecule from the lumen of the digestive system here into the cytoplasm of these absorptive cells, and then eventually moving it through the other membrane into the capillary bed that belongs in the villus here, or in some instances this lymphatic vessel that extends up into the villi, which is called a lacteal. Okay. So this picture is kind of denoting this relationship here from the lumen out, but at the top of it. All right. So what we're going to do is we're going to kind of start and go through kind of a pattern that we just talked about, the digestion. So carbohydrates, the role of the digestive system is to take complex carbohydrates and then break them down into simple carbohydrates. So the most common one would be glucose, and then we would have some galactose from lactose in milk, and we'd have some fructose from sucrose, and then there are plant material like fruit that have fructose in it as well. So glucose and galactose are actually absorbed via coupled transport, and it's going to be absorbed by a symporter system. And so we call that secondary active transport, which is kind of an old name for coupled transport. And so what we need is we need a sodium ion, and we need one of the sugars, glucose and galactose. And then we're going to move the sodium and the sugar into the cell. And then eventually, because we're absorbing it, we can create a gradient that allows us to diffuse it into the blood. So we're going to use active transport on the lumen side, and we're going to use diffusion on the other side to get it into the blood. So what's interesting is fructose is done by facilitated diffusion, which means that we would never technically be able to absorb all the fructose that's in the lumen. And that's one of the reasons why if you go to the grocery store, you can find fructose in the specialty aisle as a dietary sugar, because you aren't as effective as absorbing fructose through your gut lining, and you also aren't quite as effective as using it to make ATP for two reasons. All right. So now if we go down to lipids, which are the next group that we talked about in digestion, then what the lipase is doing is creating these monoglycerides, which is glycerol with one fatty acid, and then these free fatty acid molecules. So one of the things that happens in your gut is that because fats are not soluble in water, we end up with fat droplets forming through the digestive process that we call micelles. And because the membrane is made up of phospholipids, and one of the issues with moving something through a membrane is that we have this bilipid layer where the charged part of the phospholipid is toward the outside, which would be toward the lumen of the system. And then the other phospholipid would be toward the cytoplasm of the cell. And we end up with a membrane that has a charged entity, which we would say is hydrophilic. And then we have this area in the center, which is a non-charged area, which we would say is hydrophobic. And so it's really this part of the membrane that limits movement through a membrane based upon size and charge of a molecule. And we don't have that issue with fats. because fats will readily pass through this area of the membrane. So then it's not surprising that we use diffusion, simple diffusion, to move fats, because we don't have that problem of transport across the membrane to do that. So we actually use simple diffusion for short-chain fatty acids, and then we use simple diffusion for these micelles, which are monoglycerides and long-chain fatty acids. And then what we do is we move them into our cell. Then what our cells do, our absorptive cells, is they take the monoglyceride and fatty acids and they convert them to triglycerides. And then what we're going to do is we're going to package the triglycerides into a little packet we call a chymomicron. And then that's going to be picked up not by the capillary bed, but by the lacteal that's at the center of each villi. So that's really the critical reason why we have a lacteal, is to do these dietary fats. And then we're going to depend on our liver to process these fats so that they're more transportable in our blood. So that leaves us with our proteins. So again, we have a wide array of digestive enzymes to digest proteins. So the end product we end up with is largely amino acids, although we can get some fragments which are dipeptides 2 or tripeptides 3 amino acids held together. And then with amino acids and with these end products, we're going to do, again, secondary active transport, where we're going to move 2 molecules simultaneously. But free amino acids are moved with sodium, similar to what we saw in glucose and lactose. And then dipeptides and tripeptides are actually moved with hydrogen ions, which is kind of clever because our stomach secreted hydrochloric acid, which had a lot of hydrogen ions, and now we're going to help reabsorb some of our hydrogen ions in this process that helps us digest, that helps us absorb these protein amino acid products that we need to make proteins. So then the next step is that once we have absorbed all of these nutrients, and we either have them in our lacteal here, or we have them in our capillary bed. So at one end of our capillary bed, we have an artery that feeds the capillary bed in the villus. And then what drains the villus is a vein. And then all of the veins from the small intestines come together to form a vein that goes to the liver, which is called the hepatic portal vein. So it's called a portal vein because we have 2 capillary beds linked together. So we have this capillary bed in our villus, in our small and large intestines, then we have this capillary bed in our liver. And so the blood vessels called the hepatic portal system. So the take-home message is all of the venual blood coming out of capillary beds in our small and large intestine all pass through the liver before they go anywhere else in the body. And then that allows the liver to do its job of processing these nutrients, detoxifying things that could be ingested like alcohol or other toxins, and then processing that before it goes to the rest of our body. And so that's what we're going to talk about in the next lab, is the process that the liver does as these nutrients are brought back via the hepatic portal vein.